Bismillahirrahmanirrahim. This is the lecture number 6 in series of the photosynthesis lectures. In this lecture, we are going to discuss the C4 cycle which happens in the C4 plants, and one of the examples of the C4 plants is rice. Now, we are going to discuss the C4 cycle in detail in this lecture. Now, if you guys remember, in the last lecture, I told you that why do we call it C4 cycle, because during this reaction, the first molecule which is achieved, that is of 4 carbon compounds. Now, this C4 cycle reaction, it happens in 2 cells. The first cell in which this chemical reaction takes place, that is called the bundle sheath cells, and second cell in which this chemical reaction is completed, that is called as mesophyll cell. Now, let's suppose that this one is the bundle sheath cell, and this one is the mesophyll cell which contains the chloroplast. Now, what happens that from the outside, the carbonic side, it enters into the bundle sheath cells. Now, in the bundle sheath cell, one molecule which is already present, that is PEP. Now, we have discussed this molecule before as well in the Krebs cycle. The complete name is phosphoenol pyruvate. Now, this carbonic side joins with phosphoenol pyruvate in the presence of an enzyme, and the name of the enzyme is PEPCO. Now, this PEPCO, it stands for phosphoenol pyruvate carboxylase. Now, as you already know that this phosphoenol pyruvate, the PEP, this is actually a 3 carbon compound, and carbonic side is 1 carbon compound. When both of these, they will join together, a 4 carbon compound will be formed. So first compound formed is a 4 carbon compound, and the name of that first compound is oxaloacetate. Now, in next step, what happens that this oxaloacetate, it changes into another 4 carbon compound, and that 4 carbon compound is called as malate. Now, this reaction happens when one molecule of NADP H2 change into NADP. Now, the enzyme that carries out this chemical reaction, the name of that enzyme is malate dehydrogenase. Now, this is actually a reversible chemical reaction. For your understanding, I'm not showing it as reversible. In next step, what happens that this malate is then transported from the bundle sheath cells into the mesophyll cell. Now, in next step, what happens that this malate is then broken down into a 3 carbon compound which is called as pyruvate. Now just remember that the malate, it is a 4 carbon compound while pyruvate is a 3 carbon compound. Now it means that one carbon is decreased and that carbon is released inside the mesophyll cell in the form of carbon dioxide. Then this carbon dioxide joins with the ribulose bisphosphate in the presence of the enzyme Rubisco, about which we have learned in detail before. Then this ribulose bisphosphate, it forms the complete Calvin cycle. It goes through the complete Calvin cycle and we get glucose. Then what happens to this pyruvate? This pyruvate, it changes back into phosphoenol pyruvate. Now this pyruvate first enters back into the bundle sheath cell and then it changes back into the phosphoenol pyruvate, and while changing it from the pyruvate to the phosphoenol pyruvate, at that time one molecule of ATP breaks down to form a molecule of ADP. Now this is how this cycle is completed. Now let me tell again that from the air the carbon dioxide enters and it then enters into the bundle sheath cell. In the bundle sheath cell, phosphoenol pyruvate, which is a 3 carbon compound, is already present, and this 3 carbon compound joins with the carbon dioxide in the presence of the enzyme which is called as PEPCO, and its full name is phosphoenol pyruvate carboxylase, and a 4 carbon compound is formed which is called oxaloacetate. Now this oxaloacetate then changes into malate, and this reaction happens during the presence of the enzyme which is called malate dehydrogenase, and during this one NADPH2 molecule changes into NADP. Then this malate moves from the bundle sheath cell into the mesophyll cell, and in the mesophyll cell this malate breaks down into pyruvate. Now when this malate breaks down into the pyruvate, the malate was 4 carbon compound and pyruvate is a 3 carbon compound. One carbon which is decreased is lost in the form of carbon dioxide, and that carbon dioxide joins with the ribulose bisphosphate in the presence of the Rubisco to form a 6 carbon compound, and then 2 3 carbon compounds, and the whole Calvin cycle will take place, and at the end we'll get a molecule of the glucose. Now this pyruvate, it will move back into the bundle sheath cell and then it will be changed back into the phosphoenol pyruvate so that it can be used again in the C4 cycle. Now the main question is, when the same Calvin cycle is happening in the mesophyll cell, then why there was requirement for production of oxaloacetate and malate, and why this bundle sheath cell has to take part into this chemical reaction? The answer is very, very simple. Now these C4 plants, they reside into the hot environment, and usually these crops, they are produced during the summer season, and during that time what actually happens, the carbon dioxide concentration becomes less because of the expansion of the air. And when the carbon dioxide concentration becomes less, so at that time the Rubisco, instead of performing carboxylase activity, it can perform oxygenase activity, which can be loss of ribulose bisphosphate, which will be loss of photosynthesis. So to increase the concentration of the carbon dioxide in the mesophyll cells. So what happens that first of all phosphoenol pyruvate joins with the carbon dioxide to form oxaloacetate and then malate, and then these malate first it increases its concentration in the mesophyll cell, and then when it becomes to the higher, reaches to the higher concentration, then malate breaks down into the pyruvate. So inside the mesophyll cells the concentration of carbon dioxide remains higher. So the C4 cycle is actually for the prevention of the oxygenase activity of the rubisco, so that there should be no loss of photosynthesis. So this is how the C4 cycle helps the C4 plants. So I conclude my lecture over here. See you guys in next lecture. Thank you very much.